Hi students, when we left off I was talking about glycolysis. It's the first step of cellular respiration. Now the next part of cellular respiration involves the mitochondria. The mitochondria accepts the pyruvate molecules, but something has to happen first. The pyruvates will enter the mitochondrial matrix as a 2-carbon acetyl group. Okay, they attach to an enzyme called coenzyme A to form acetyl-CoA. When this happens, an electron is picked up by NAD+, and carbon dioxide molecule is released. The carbon dioxide released at this step is, for humans, transported out of the mitochondria, diffuses through the cytoplasm to the membrane, diffuses across the membrane, and goes into our bloodstream to be exhaled. This is an interesting place where we can kind of picture what's going on at the cells affecting our overall organism as a person. So where does the glucose come from? It comes from food that we eat. And the glucose combines with oxygen, which will happen later on. We'll see the oxygen. But for now, the glucose is being broken down. Some CO2 gets released. We've gotten a little bit of ATP from this process. If you remember, 2 molecules of ATP were produced in glycolysis. So now we have our molecule ready to enter the citric acid cycle. Here's just a review of the mitochondria, and you can see the double membrane here. The cristae are the infoldings of the inner membrane, and the matrix is actually a fluid-filled space that contains ions, enzymes, and other molecules that function in cell respiration. At the bottom of this slide, you can see an actual electron micrograph of a cross-section of a mitochondrion. So this part of the process that I just described is sometimes called the transition reaction or the preparatory reaction. And that's when the pyruvate gets added to coenzyme A, it gets oxidized, electrons are removed, becoming acetyl-CoA and 2 carbon dioxides are produced. So this isn't a step of cell respiration. Your book describes the 3 steps, but this is a transition between what occurs in the cytoplasm and what occurs inside the mitochondria. The citric acid cycle is often called the Krebs cycle for Hans Krebs, the scientist who discovered the process. And in the citric acid cycle, the acetyl groups received from that preparatory transition reaction go through a cycle of reactions. You're going to see on the coming slides that this is a complex process involving several intermediates. Please look at your textbook on the illustration of this so you can see how complicated it really is. I don't expect you to remember all of the intermediates, but I do want you to know 2 of the molecules in the cycle. And the first one is oxaloacetate. It's hard to pronounce, but this is an important molecule because it picks up the acetyl-CoA, bonds to it. Okay? Oxaloacetate bonds to acetyl-CoA, and this forms citric acid. That's the other molecule I want you to remember. Citric acid is sometimes called citrate, same thing. So oxaloacetate plus acetyl-CoA becomes citrate. And then this citrate gets broken down over and over, releasing a little ATP, releasing electrons, releasing CO2, until it ends up being oxaloacetate again. Therefore, it's a cycle. Oxaloacetate bonding to something and being broken down back to oxaloacetate. Then the oxaloacetate can bond to another acetyl-CoA, and the cycle can continue. So electrons are removed. NAD+ and FAD take those electrons. I have here NADH and FADH2 carry those electrons or capture them. So those NADH and FADH2 are the reduced forms carrying the electrons. The electrons will be used in the electron transport chain next. 2 ATPs are formed, 1 for each acetyl-CoA that goes into the cycle. This illustration shows that cycle occurring, and there's a very similar picture in your textbook. So I'm looking at the purple cycle down at the bottom of the slide, And to the left of it, you can see the acetyl-CoA. It bonds with the oxaloacetate, making citrate. Then we could see the series of reactions that oxidize the citrate and release carbon dioxide. At the end of this cycle, the original acetyl-CoA is broken down completely, and really we can think of this as the completion of breaking down the glucose. So nothing from the original glucose is present anymore. The CO2 gets exhaled from our lungs, or it diffuses out of the cell, and the electrons will be used in the electron transport chain along with the hydrogens. So here we've completely broken down glucose, and the only thing that needs to happen next is to get the rest of the energy from it. So far we only have 4 ATP molecules, and cells need typically more energy than that. At least our human cells will need a lot more energy than that to do what they need to do. So in the citric acid cycle, 2 acetyl groups come in. This carbon in the acetyl groups and oxygen gets released as carbon dioxide. You don't need to know the numbers of NAD+ and FAD involved, but just know that they're both involved here as electron carriers, and we make 2 ATP molecules. So the final step of cellular respiration is the electron transport chain. And this occurs on the matrix, or in the cristae of the mitochondria, just the border of the matrix. Okay, the cristae is that infolded inner membrane. Now the electron transport chain is a series of molecules that need to be bonded in a certain order to take electrons. These molecules of the chain need to be on a membrane. And so eukaryotes have this in the mitochondria, but what about those prokaryotes, the archaea and the bacteria? Well, they don't have mitochondria, so their electron transport chain is located on the plasma membrane. The molecules of the chain are proteins and cytochromes. Cytochromes are respiratory molecules that often have pigment to them. The pigment is often a reddish color because the metal atom that's in the center of the cytochromes is usually iron, and it gets oxidized. Oxidized iron is rust, and so we often see the mitochondria portrayed as a rust-colored organelle. So the electron transport chain accepts the electrons from NADH and FADH2 and carries those high-energy electrons down the chain to a low-energy state. This will help the production of ATP. In this slide, you can see some of the molecules involved in this chain. Fe is iron, S is sulfur, CYT stands for cytochrome, and you don't need to memorize the organization of the chain. But you can see here that we start with electrons as 2 NADH, that's carrying our electrons, and they're at the high energy state at the top. Down at the bottom, you can see just barely, because my picture is in the way, the oxygen is at the very bottom. Oxygen is the final electron acceptor in the electron transport chain. And here, the oxygen with the electrons will combine with hydrogen. Remember, electrons and hydrogens like to travel together. Now, what does hydrogen and oxygen make? Water. So there's our final product of water. This is another illustration showing the electron transport chain. And here you can see the molecules embedded in the cristae of the mitochondria. The electrons are shown as blue spheres. The proteins and cytochromes are those big orangey blobs. And the NADH is carrying the electrons to the electron transport chain. You'll notice that something else is happening in this picture, and you'll read about this in your textbook as well. Hydrogens get pumped across the membrane as the electrons are passed down the chain. What this does is form a concentration gradient of hydrogens on both sides of the membrane. We end up with a high concentration on one side and a low concentration on the other side. Now, what happens when we have that concentration difference? Diffusion. The hydrogens want to diffuse across the membrane, and they can do so only through one molecule called ATP synthase. As the hydrogens diffuse across the ATP synthase makes or synthesizes, you guessed it, ATP. So here's where we get the production of ATP. And we get a lot of ATP from this. We get about 32 to 34 molecules of ATP in the electron transport chain. So we get a max total of 36 ATP molecules here. When you see total molecules of ATP produced through cellular respiration, it's going to be in the 30s. Now just look at how efficient this is. We have our reactant glucose, and that has a certain amount of energy to it. If we have 1 mole of glucose, that is a certain number of molecules of it, a mole has 686 kilocalories of energy. The yield is 36 ATP molecules, and that is 263 kilocalories. This can be measured using a calorimeter. So the efficiency here is only about 39%. The balance of the energy is lost as heat, and that follows the second law of thermodynamics, where for every energy transfer, some usable energy is lost from a system to increase the entropy of the universe. So the waste heat increases the entropy. This slide shows an overall energy yield per glucose molecule, and it kind of summarizes the different parts of the process. So take a few minutes to look at this slide and just notice where the electrons are, where the parts of glucose are, the actual substance of the glucose molecule, and where the processes are taking place. Now I'm going to end this video, and the next time you see me, I'm going to talk about anabolic and catabolic metabolism and fermentation.